Hello everyone, today my topic for presentation is enzymes. My name is Ayush Mahendra, roll number is 043. So let's start. So the content for today topic is what is enzymes, classification of enzymes, isozymes and active enzyme sites, Fisher's laws and key models, Coastline's induced fit models, factors affecting enzymes unit, enzymes inhibitions. So let's start. What is enzymes? Enzymes are the biocatalysts. They catalyze the biochemical reaction both in vivo and as well as vitro. So they are highly specific to its substrate and have great catalytic power. That's why they enhance the rate of reaction tremendously without being changed. There are many enzymes which requires cofactor for their catalytic activity. The cofactor may be a complex organic molecules called as coenzymes, or it may be a metal ion such as Fe2 plus Mn2 plus, and enzymes plus its factor is called as holoenzymes. In such cases, the protein component in cofactor enzymes is called apoenzymes. So let's start with classification of enzymes. So basically there are six types of enzymes. First is oxidodactases. So basically it is a oxidation reduction reaction where electrons are transferred. Second is transferases, where transfer of groups is done. Third is hydrolysis, a hydrolytic reaction is done. And fourth one is lyases, addition or removal of group to form double bond. And fifth one is isomerases, transfer of groups within molecules to yield isomeric form. And sixth one is ligases, condensation of two molecules coupled through ATP hydrolysis. So they are the classifications of enzymes. So next is isozymes and active enzyme sites. So many enzymes are present in multiple form in the same species, tissues, and even in the same cells. These enzymes are called isoenzymes or isozymes. And the enzymes active sites. The catalytic reaction performed by enzymes occur at particular site on the enzymes. This site is called as active enzymes. So the active sites is clearly defined a pocket or cleft in the enzymes molecule where the whole or a portion substrate can fit. So next topic is Fisher lock and key models. In 1894, the introduction of lock and key model for the substrate and enzymes interaction was proposed by Emil Fisher. According to this model, complementary structural features are present between enzymes and substrate, and the active site is pre-shaped to its fit substrate. The substrate can fit into the complementary sites on the enzymes as a key fits into a log. This result in the formation of enzyme substrate complex. So this is the Fisher lock and key model. We can see in the figure that how it perform. And next model is Coastline induced fit model. In this model, Daniel Coastline in 1958 proposed induced fit hypothesis. He just suggested that the structure of substrate may be complementary to that of active site in the enzyme substrate, complex but not in the free enzymes. The interaction between the substrate and the enzymes induces conformational charges changes in the enzymes, which align the amino acid residue or other groups for substrate binding, catalysis, or both. So in figure we can see that how substrate and enzymes are different, but after a Coastline induced fit model they together form enzyme substrate complex. So next topic is factors affecting enzymes activity. There are various factors that affect the enzymes activity. First one is temperature. We can see in the graph that the rate of an enzyme's catalyst reaction increases with the increase in temperature up to the maximum and then falls. When a graph is plotted between temperature versus enzymes activity, a bell-shaped curve is obtained. We can see in the first figure. The temperature at which the maximum rate of reaction occurs is called as enzyme's optimum temperature. We can see that before 40 it has optimal temperature. And next factor is hydrogen ion concentration. So enzymes activity is also affected by pH. A plot of enzymes activity against pH result in a bell-shaped curve. We can see in second diagram that each enzyme has a unique optimum pH at which the rate of reaction is greatest. The optimum pH is the pH at which the activity of particular enzymes is at maximum. More enzymes of higher organisms show optimum reaction rate and around neutral pH around 6 to 8. And the third factor that affect the enzyme's activity is substrate concentration. In the graph again we can see between rate of reaction and substrate concentration that the substrate concentration also influences enzyme's activity. As the substrate concentration increases, the rate of reaction also increases. This is because the more substrate molecule will interact with enzymes molecule, the more product will formed. However, after a certain concentration, further increase in substrate concentration will also no effect on the rate of reaction. We can see in the graph that after some further time, there is no change in the reaction. Since the substrate concentration will no longer be limiting factor, at this stage enzymes molecules become saturated and work at their maximum possible rate. Next topic is in enzymes unit. The enzymes unit is the amount of enzymes that catalyzes the conversion of 1 micromolecules of substrate per unit per minute under standard condition. The International Union of Biochemistry adopted enzymes unit as a unit of enzymes activity in 1964. And next topic is enzymes inhibitions. In these substances which decreases the rate of an enzyme's catalysis reaction are called as enzymes inhibition. So there are the two types of inhibition: reversible inhibition and irreversible inhibition. Irreversible inhibition: the inhibition binds very tightly to the enzymes and does not dissociate from it. For example, the antibiotic penicillin act as an inhibitor and bind with the enzymes transpeptides, which is responsible for synthesis of bacteria vessel wall. This reversible inhibition are of three types: competition, competitive, non-competitive, and uncompetitive inhibition. So here we can see that the types of inhibition: reversible inhibition, competitive, uncompetitive, and non-competitive. So firstly, competitive inhibition. In competitive inhibition, there is a close resemblance in the structure of inhibitor and substrate. Therefore, they both compete for some same active time on the enzymes. We can see that in the figure how they work at the same time on the enzymes. Now the second one is non-competitive, uncompetitive inhibition. In this type of inhibition, inhibitor does not bind to the free enzymes, it binds only to the enzyme substrate complex directly on its binding is facilitated by the conformation changes that take place. And third one is non-competitive inhibition. In this type of inhibition, we can see in the figure that it has no structural similarity with substrate and binds with the enzymes at different site other than the active site. Therefore, this is non-competitive between S and I, which is substrate and inhibition, and the formation of ES and EI and ESI take place. So this is all about types of reversible inhibition. That's all about enzymes. Thank you everyone.